there listening to this, wondering where your next grant's going to come from, you can just hear them whining away. No money, no money. But they're all wine-like in nature. Now, if we look at the amphibian papilla, the tuning of the amphibian papilla in all of these species, the tuning of the amphibian papilla, the AP, matches the dominant frequency of their whine. What about the chucks? Well, these guys don't make chucks. But they still have a basal papilla, and every sensory end organ is tuned. All sensory end organs are nonlinear. So what we see here is that with one exception, this other thing called pustulatus, this has a higher tuned basal papilla. For the rest of these species, there has been no evolution of tuning in the basal papilla for at least 15 million years. So what that tells us is that the tuning of the basal papilla that now matches the dominant frequency of the chuck, both in pustulosis and in Peter's eye, the tuning of that end organ existed long before males ever evolved these chucks. So what that suggests to us is that the female auditory system and their preference for complex calls did not co-evolve with the complex calls. Instead, that females had instantiated in their auditory systems these sensory biases, these tuning biases. When males evolved chucks, they evolved chucks that we assume it was a gradual process that slowly maximized their stimulation of this female's end organ. And these are the data that led us to suggest this idea of sensory exploitation, that in some cases in sexual selection, males evolve traits that exploit these pre-existing perceptual and cognitive biases in females. So that kind of gives us the background that we need to know to understand why we move to this next level of analysis. So this next level of analysis is still asking about mechanisms, but it's not taking a bottom-up approach from the auditory system in the brain to behavior. We're concentrating on the behavior now and hoping that at some point we can return to the brain and look more specifically at some of these neural mechanisms that are generating some of these cognitive biases. So I want to talk about a few of these. We've been studying a list of cognitive biases in these animals, and I don't have time to talk about all of them, so I want to concentrate on two things. One are studies asking how animals compare stimulus quantity, and the other ask how animals deal with communication systems that are perceived through more than one sensory modality. So I would like to know how animals compare quantity. When we think about sexually selected traits, like a peacock's tail, what we usually see is that sexual selection results in the elaboration of traits. In 1992, Ann Kitty Hector and I reviewed a number of studies of sexual selection, about 100 or 150, and what we found is when there is evidence for female preferences within a species, the females usually prefer the traits of greater quantity. Calls that are louder, calls that are longer, calls that are more complex, colors that are brighter, ornaments that are bigger, that are larger, that are longer. There's exceptions to this, but in more than 90% of the cases, there's preference for the more elaborate. And it's the preference for the more elaborate that caused Darwin to get sick when he saw the peacock's tail, because if you've ever seen a peacock displayed to a female, it's a beautiful thing to watch. I'm sure it's a beautiful thing for the female to watch, but did you ever see a fox or even a kid chase a male peacock? You know, it's pathetic. They can barely get off the ground. They can barely fly. This is the cost of sexual selection. That not only are these sexually selected traits that females find attractive, but they're often traits that are very costly to the males. So if there's selection for more, how much is more? How much more is enough? So we want to ask ourselves a really fundamental question about how animals compare stimulus quantity. And we can get an insight into that by asking how we compare quantity. So what I'm going to do is I'm going to show on the screen two clouds of dots. And I want you to raise your right hand if you think there's more dots on the right-hand side, and your left hand if there's more dots on the left-hand side. But you can't cheat. And what's cheating? Cheating is counting. So this has to be Gestalt. Okay, so I'm going to put up this slide, and then immediately you have to raise one of your hands. Okay? Ready to go. Okay, either you all got it right or you all got it wrong. You all got it right. Okay. Now that wasn't hard. Everyone got it right and latency was really fast. What was the difference here? There was one more dot on the right, and that represents a proportional difference of 20%. Okay? Let's do it again. Ready? Most of you, not you, most of you got it right, but the latency was substantially longer. Okay? What are we seeing here? Again, there's an absolute difference of 1, but now the proportional difference is only 0.08. One more time, ready? Okay, so it's still on the right, is the most dots. And what we see again is that the absolute difference is 1, but now the proportional difference is very small. It's only 0.02. So this psychophysical phenomenon was discovered by Weber in the late 1800s, and then it was also studied by Fechner, and this is referred to, this type of comparison that we're doing is called Weber's law, or sometimes it's called Weber-Fechner's law. And what this says is that Weber and Fechner were interested in how different do stimuli have to be for us to tell that they're different from one another. And in psychophysics, the term that they use is a just noticeable difference, or a JND. So what's the minimum difference needed for a JND? And Weber's law says that there will be a constant that you need to be able to discriminate between two stimuli. And that constant is delta I, the difference in the stimulus magnitude between two stimuli, divided by the absolute magnitude of the stimulus. So what that means, and which is important for sexual selection, what that means is that as stimulus magnitude increases, the difference between stimuli has to become greater with each increase in magnitude for individuals to be able to distinguish them. Now, this idea was applied to sexual selection in, I think it was 1982, in a paper in Ethology by someone called Cohen, C-O-H-E-N. And the title of the paper was The Psychophysics of Mate Choice. And when I was at Berkeley, a very well-known ethologist, George Barlow, came to my office, and he said, "Mike, you have to read this paper." So I read the paper. I was incredibly excited about it, because this guy, Cohen, I want to make sure he gets the credit, not me, I didn't think of this. Cohen said, if Weber's law is acting in sexual selection, that can have some really important influences on the dynamics of trait evolution. It might lead to, these are my words, not his, it might lead to a cognitive break on the evolution of elaborate traits. So that was in 1982. So then about 20, 25, 30 years later, we decided, whoa, why don't we test this? So before we started to test this, I said, I'm going to look at the citations for Cohen's paper, because I know it's never been tested in mate choice, but I want to just be sure, so I could see who all cited his papers. Plus, it was such an amazingly interesting paper. I'm sure it's been cited tons of times. So I go back, just as we're doing this study, and it had been cited, I think, 12 times. And 8 of those were by me, and there's no data. So and I've never met Cohen. So anyway, this idea goes back to this theoretical paper that was done in, obviously wasn't paid much attention to for many years. So what we wanted to know is that with chuck number, females prefer more chucks to fewer chucks. How are they making these comparisons? Are they comparing absolute differences, or are they following Weber's law, which would mean they're using proportional differences? So we can test this hypothesis. Why are you not moving? Did it move? Yes. Okay, so we can test this hypothesis by giving females pairs of calls that vary in the number of chucks. If females are using absolute differences, then the strength of the preference for, let's say, 6 versus 5 chucks should be the same as the strength of the preference for 2 versus 1 chuck. If they're using proportional differences, then there should be a much stronger preference for 2 versus 1 than there would be for 6 versus 5. So we tested females with a lot of combinations of pairs of calls that varied in the number of chucks. So here are the results. Well, first I should point out, our proxy for the strength of preference is a population proxy. So in these studies, the sample sizes, I forget, they're somewhere between 30 and 50. So the proportion of females that preferred the call with more chucks is the strength of the preference for that call. So that's what we plot on the x-axis. On the y-axis on the x-axis in the top graph, we plot the difference in the number of chucks between the two calls that the females chose between. And as we see here, the R squared is only 0.12. So even though females preferred calls with more chucks to fewer chucks, the absolute difference doesn't explain a lot of the variation in the female's preference. Well, when you add chucks, you also influence the amount of energy in the call, and for some subtle reasons, it's not always linear. So this is another kind of independent variable. So if we look at the amount of energy difference between the calls that varied in the numbers of chucks, again, the R squared is only 0.16, so it's not explaining a substantial amount of the variation. But if we plot the ratio of the chucks in the two calls that the female is presented with, then we see that the chuck ratio explains a substantial amount, 84% of the variation in the strength of female preferences. So the females do seem to be following Weber's law when they're making this comparison. Why? Well, there's two general hypotheses here. One hypothesis, we just refer to as cognitive constraints, is the females are doing this because this is what vertebrates do. This is the way comparisons of magnitude are done in vertebrate brains. Frogs are vertebrates, so this is what the Túngara frogs do. The other explanation is a much more specific one, and that's some kind of an honest signaling hypothesis. It's possible if chuck number, we don't have any evidence that it does, but if chuck number was indicative of some aspect of male quality, where the more chucks that are produced, the higher the male quality in terms of survivorship, not in terms of sexual attractiveness, because we know that that's true. It might be that the difference between males making 6 chucks versus 5 chucks in quality might be less than the difference of males making 2 chucks versus 1 chuck. So we tested this hypothesis in a number of different ways. We looked at the relative condition of males' propensity to make chucks, and we find no relationship. We looked at the male's body mass, we found no relationship. We looked at the male's size, we found no relationship. And the problem with, the good thing about studies of adaptations is we can make very precise predictions that are testable. The tough thing about finding negative data for adaptation is it's always possible that you haven't been able to identify the adaptation. So we thought maybe the way to crack this, because we tested all the adaptational hypotheses that we had access to. So we thought, well, maybe what would be great is if we had another animal that we could ask to count chucks, and we could ask that animal if they were using Weber's law or making absolute comparisons, and if this other animal didn't care about mate quality whatsoever. So lucky for us, we have just the perfect candidate for this. So this is the frog-eating bat, Trichopterosis, and Merlin Tuttle and I, when I was a graduate student, we discovered that this bat not only eats frogs, but it finds the frogs not with its echolocation system, but by passively localizing the calls of the frog. In a follow-up study with some neurobiologists in Eastern Europe, what they showed is that these bats, tricops, has a series of adaptations in the inner ear that extends their frequency range down into the realm of where frog calls are. Their echolocation signals are about 60,000 hertz, and most bats are not very sensitive to our sonic sounds, but these bats they extend the basal membrane into a region that's sensitive to sounds below 5,000 hertz, where most of the frogs are. So we know that tricops will go to these calls. They respond to the calls very much like female frogs do. If you present a tricops with a whine-only call, the male will fly either out of the canopy or from his perch in a flight cage and come down and make passes over the speaker, sometimes landing on the speaker, trying to get to the frog inside. So a whine is sufficient to attract a frog-eating bat. And like the females, they prefer calls with more chucks to fewer chucks. So we did these same experiments with tricops, and we asked how the tricops are exhibiting their preferences for more chucks versus fewer chucks. And to make a long story short is that they're also doing these Weber function-like comparisons, shown by the red line here. The absolute numbers of chucks, the energy differences in the amount of call goes nowhere to explaining the variation in the bat's behavior. And this Weber function of the bats is statistically indistinguishable from the Weber function of the frog. So we want to now go to a different sensory modality, and that is the combination of visual and auditory cues. Now, we know that when we speak, our lips move. We know that people can extract information about the spoken word by looking at our lips. Deaf people can do it quite well, but we can do it well. If you're talking to someone, especially in another language, and you're having trouble hearing what they're saying, you usually stare at their lips, and it helps you. It gives you more information. But the movement of the lips is just an incidental consequence of our vocal production system. Well, there's something analogous in frogs. They have a vocal sac. The vocal sac evolved to cycle air back and forth so it can be used again and again and again in producing calls. Frogs don't have a diaphragm. They don't have positive pressure like we do. They basically have to swallow air and force it into the lungs. And it's something that takes a good bit of time. And we've shown that when the frogs have to do this buccal pumping, then their call rate decreases. And we've also shown that females prefer faster call rates. So what we wanted to know is this inflating and deflating vocal sac a part of the courtship display that females recruit when they make decisions about with whom to mate? So with Ryan Taylor, who was a postdoc in my lab at the time, and we continue to work together, we constructed this robot frog. And the robot frog has an inflatable vocal sac that's driven by a pneumatic device that's slaved to the computer, and the pneumatic device follows the waveform of the call. So as we see in these lower pictures, the inflation cycle of the robot is a pretty good match to the inflation cycle of the males. In a public service announcement, when we first made these robo-frogs, we used condoms, and they broke immediately. So we had to switch to catheters, catheters, which last a lot longer. So do what you want with that. Okay, so these experiments are pretty simple. We give the female a choice between two calls, one from each speaker, but one of the speaker has a robo-frog underneath, in front of it. And we put the speakers at this angle so the females have visual access and auditory access to both stimuli at the same time. This is done under half-moon conditions in the field. So both the amount of illumination and the spectrum of illumination is the same as the moon. And we thought that would be hard to replicate, but if you go to a store and buy moonlights, that you plug in, little dim lights, and if you measure the spectrum of those moonlights, amazingly, it's exactly what the moon looks like. So I never would have thought that they would have cared. So what we do now is we say to the female, if you can hear a call, and if you can hear and see a call, do you have a preference? Well, one possibility is the females are just going to get freaked out by this robot, but that's not what happens, as you see in this video. Now remember I told you earlier that the female makes physical contact with the male to choose him. And in this species, like most species, males don't turn down these kinds of sexual invitations. So the female here is very persistent in trying to be chosen and is not chosen by the robot. There's some simple experiments. We've worked with these Túngara frogs for a long time, but what we do, when we start a new series of studies, we always go back and we redo the experiments that we did years before, especially when there's some change in the testing paradigm. So in this experiment, we moved the speakers at a different angle. So we redid this experiment, wine chuck versus wine, and we get the same results, like we always get. Females show a very strong preference for wine chuck versus wine. Now, if we do wine chuck versus wine chuck, but the wine chuck has the robot, what we see is the females like the robot. They show a very strong preference for the visual plus acoustic stimulus over only the visual stimulus. Now, with this phenomenon of reading lips and hearing voices, McGill published a very famous paper in the 1970s in something that's been known as the McGill effect. And if you change the pattern at which the lips move, and there's lots of YouTube videos showing this, if our lips say ba, but the voice coming out of the video says pa, then we hear da. And then if you slowly morph the movement of the lips to what would normally coincide with that phoneme, then very quickly we can hear that phoneme, very quickly. And so it's also used as a demonstration of categorical perception. So this is what we wanted to know. How important are temporal relationships of components in the auditory realm and also in the audiovisual realm? Well, they're important in the auditory realm. If we move the chuck, so there's a 500 millisecond gap, so instead of the females hear and if we give them a choice between this call with the temporally displaced chuck and a whine, then the females don't discriminate. So to them, the chuck is not part of the signal. And a big influence on a lot of these kinds of studies for me is from Bregman, who was a faculty member here and wrote this amazing book on auditory scene analysis. And using the lingo that Bregman would use, he would say that there's no perceptual binding of this chuck to this whine. So there's no continuous perceptual stream, so the females don't put these two together in their brains. What about the visual signal? Well, the same thing happens with the visual signal. If we move the vocal sac inflation to occur right after the chuck, then it no longer adds to the salience of the signal. In fact, it makes the signal significantly less attractive. So what we see here, there's a real premium on the temporal relationships of the acoustic components and the visual component with the acoustic component. Then what we wanted to know is, what about if we were to take the frog and we were to shove it in this gap here? Now, Bregman, in his book, spends a lot of time talking about a really interesting auditory phenomenon called auditory continuity. So if we were to hear two tones, same frequency, we can tell they're two tones. Now, if you put white noise in between those two, so two distinct tones. As you increase the level of the white noise, the tone becomes continuous. And it glides right through the noise. So Bregman pointed out this phenomenon, and again called it auditory continuity. We wanted to know if there is a more complex kind of continuity, a multimodal or perceptual continuity. So we did this experiment. We asked, well, how would the female respond to this signal versus this one, which she treats just as a whine? And what we found is that when we put the visual signal between these two signals, which never happens in nature, it instantiates the salience of the chuck into the signal again. It makes it more attractive than this dissociated call. How much more attractive does this make it? Amazingly, to me at least, amazingly, it makes it just as attractive as a normal whine chuck. So what this says, the kind of the take-home message for this kind of study is we tend to think of these signal and receiver systems as not only tightly co-evolved, but tightly knit together like a lock and key mechanism. What we're seeing here, though, is that there's this amazing permissiveness in what the females are finding attractive. Now, males aren't going to evolve a call where they make the whine and then they inflate the vocal sac and then they make the chuck. But what this says to us is that there is this huge perceptual landscape of unexplored preferences. So when changes in signals, when mutations in signals occur, it's very possible that females might find them attractive without any coevolutionary change on the part of the receiver. There's many other phenomena, such as peak shift displacement, which happens with zebra finches and other animals that seem to be able to exploit these kinds of cognitive processes. Okay, the last thing that I want to talk about, how am I doing on time? Am I way over? Okay, you tell me when to stop and I'll stop. Okay? So when we communicate, what we're doing is we're disturbing the environment, right? When we make a sound, our vocal folds move and we change the pattern of condensation and rarefaction of molecules around our mouth. And this change in sound pressure passes out all around us, and it makes these little membranes in our ear move back and forth and cause neural discharges in our inner ear and gives us percepts of language, of sound, of intention, of motivation. When we make a visual signal, we take available ambient light and we bias which parts of that light are going to bounce off any kind of visual signal that we're using. Well, there's something else so the frogs are doing both of these, but there's something else that they're doing. Because they call in the water and because they have vocal sacs, they make these ripples all around them. Any animal that's going to have any kind of activity on the surface of the water is going to disturb the surface of the water. So I'll show you a video of a frog calling, and we'll be able to see, you have to look carefully, but you'll be able to see that there's, that he's generating these water ripples. Okay, so we wanted to know if this is a third modality that's involved in the communication system. So with the postdoc, Wouter Hafwerk, and Wouter got his PhD at Leiden, and he worked with this ornithologist Slabberkorn. And Slabberkorn is very well known for almost starting the field of urban noise and its influence on avian communication. And Wouter's done a lot of excellent work in that area, but he's been doing some really exciting work with us on the Túngara frogs. So again, we wanted to know if the frogs were attending to these water surface ripples. So instead of using female preferences, which we'll do, it's a little more challenging, and we just did this work last year, we did experiments with males where we played male signals from a speaker, and then at the same time, we had a device that disturbed the surface of the water. We measured, after we got back to Austin, we repeated these experiments, and using a laser vibrometry setup, we measured the amplitude and the frequency of the water vibrations of this device and compared it to a real frog, a real Túngara frog calling in water, and they're pretty similar. So all we wanted to know here was, do the frogs respond differently to sound plus water ripple than to sound by itself? So what we did in the experiment, and we put the male in a mesh cage, and it'll look like he's sitting in a Tupperware box, but you're looking at the bottom of the Tupperware box, and there's just mesh that comes up. And we'll see a video of this, and what we're going to see is that the males, we're going to hear a call, and he's going to hear 1, 2, 3, 4, 5 calls that have no ripple associated with it. And then I believe it's the 6th call, we then produce the ripple, and this is what happens. So here's the male here. The speaker, the call's coming from here, the ripple will come from here. Look for the ripple. Right away, as soon as he gets the ripple, he starts to call. And what's interesting is he goes right to making 2 chucks, which we really don't see in the wild that often. And what the results show very clearly is that the males respond more to calls with chuck, to calls that have ripples associated with them than to calls that don't have ripples associated with them. Now, as I've already talked about, there's eavesdroppers that have a strong influence on this communication system. And these ripples can also be, should in theory be perceived by bats. There's a species of bat that's been very well studied, Noctilio, a fish-eating bat. And these bats fly over the water surface and they can detect the fin of a fish breaking the water surface or the ripples that are made when a fish goes through the water surface, and then they can come right down and capture the fish. So this is something that bats can do. They can be sensitive to water ripples. So the first question was, are the ripples private? And there's no reason to think that they are. Then we did insonification experiments, just basically showing that if we play ultrasound 2 waves and record the returning echolocation signal at where the bat is, that there is information there that the bats can use to locate the water ripples. So in theory they should be able to do this, but it's logical that they should be able to do this. We don't know if it's biological. So we did a very similar experiment. We had a setup where the frogs were on top of a speaker, and then underneath the frogs we had this device producing water ripples. And then we asked the bats the same question that we asked the frogs. Are they more attractive to the calls that are associated with ripples as opposed to the calls that are not associated with ripples? And here you'll see the response of the bats. And what we see here, again, is that just like with the frogs, the bats are much more responsive to calls associated with ripples and calls that are not associated with ripples. Then the last thing, I'm not going to show any data, but just to tell you the very quick story, is the frogs are able to get a lot more information out of these. If there's a thunderstorm, and if we want to know how far away the thunderstorm is, what do? When we see the lightning, we count. And we ask, how long is it before we hear the thunder? Light is